take it away. Jens, welcome.
Well, good afternoon to everyone. It is early morning in Germany. I just had my breakfast, and I understand it's sort of mid-afternoon in your time. So it's a pleasure to talk to you and to give you some of our current results on biennial bearing. I like to acknowledge my co-author Henrik Flachowski. He's from the Fruit Breeding Institute in Dresden-Pillnitz. This is the only fruit breeding institute in Germany. Now, I think it is important to very briefly define biennial or alternate bearing. Biennial bearing is the annual cyclical change in cropping behavior characterized by an on and off year with heavy and light fruit loads, respectively. So in 1 year, we might have a very intense flowering. This is followed by heavy initial fruit set, where we have very few resting spurs, and consequently, we believe this is leading to a very small flower induction. In the subsequent year, there is a very low crop load, very few flowers are initiated, and we have a very low crop load. So this is what we understand currently under biennial bearing. It can occur within a tree, within an orchard block. The occurrence of biennial bearing is very different. Now, if we start here, you may see the cursor, which I'm just moving. If we start with a competent bud, this competent bud can either maintain as a vegetative bud, or it can be transformed into a flower bud. Now, obviously, when that occurs, we call it an on-year. When the competent bud stays vegetatively, this is called an off-year. Now, to make the transition from a competent bud into a floral bud, the bud needs to undergo 3 distinct phases. First, the induction, and this is where very specific changes at the molecular and biochemical level occur. The second phase is the initiation, when distinct morphological changes occur. For example, here the doming of the meristem. And finally, we have the differentiation process, where fully a flower starts to develop over time and is ready to flower in the following spring. I think it's important to mention that most of those phases, from induction through to differentiation, occurs in the previous cycle, in the previous year of the current production year. Now, flower induction, I will mostly refer in my talk to flower induction. And in apple, this is triggered by a range of environmental cues from the literature and from our own work. We know that day length is involved. Apparently, more than 14 hours is sort of a key trigger for flower induction. Further, around 39 to 53 days after full bloom, flower induction occurs. This coincides with about 9 leaf primordia being laid down inside the bud, inside the meristem. Also, light intensity seems to be very important. The light level that the developing bud is exposed to. Equally important are a range of cultural interventions. For example, fruit thinning. I will get back to this in a minute. Applications of plant growth regulators, pruning, shoot formations, all those practical means influence have an impact on flower induction. Now, what happens with both groups, environmental cues and cultural interventions? They have an impact on a range of plant signals like genes or the proteins they encode for carbohydrates and phytohormones. And only if we have a very good constellation of all those factors, the transition from a vegetative bud into a floral bud can occur, and finally flower can happen in the following spring. Now, this is sort of the basis of molecular and physiological control of flower induction. We are very much interested here what happens inside the bud. And what happens inside the bud and whether or not the transition from a vegetative to a floral bud occurs depends on many factors. Firstly, on carbohydrates. This is obviously related to the process of photosynthesis, the leaf net carbon exchange. It is also related to cytokinins that move up from the root system in the xylem with a transpirational stream. Seeds inside the fruit are rich sources of gibberellins and auxins. And we believe the subtending bud below the fruit is very much affected by the gibberellins and auxins coming out of the fruit. And then obviously a range of genes are involved. We call them integrator genes and floral meristem identity genes, and they all positively affect the transition from vegetative to a floral meristem. However, there are also repressor genes, and I name here two repressor genes, and they inhibit the high expression of those earlier mentioned integrator and identity genes. Now, I like to concentrate on those genes that are put in bold. First, I will talk about the FT gene. From all the work in the literature and our own work, we know that FT is absolutely critical for making the transition, for triggering flower induction. When we express the FT gene from apple in Arabidopsis, you see we get very early flowering soon after we sow the seeds. But when we overexpress the FT gene from apple, then we induce again early flowering in apple. On the contrary, the FT gene is responsible for very stunted growth of the apple plantlets. They have a very distinctively different growth characteristic compared to plants where FT is not overexpressed. Now, what we tried is the FT protein to apply in under field conditions. However, that was not successful, and we could not induce flowering by FT targeted applications in the field. The other integrator gene, full, again, we believe that this gene is responsible for triggering flower induction, and we have a gene from poplar, and if we use this gene in tobacco and birch, we get very early flowering. But when we overexpress this gene in poplar, then this will not induce early flowering. However, when we induce early flowering in apple, it seems to be that the full gene is again a good candidate gene for making the transition from a vegetative to a floral bud. The fruit we're harvesting of those transformed plants, they have a normal fruit, and they have also fertile seeds, which is important when we think about those sort of techniques. Now let me talk about the repressor gene TFL1, and TFL1 activity. If this is inhibited in apple or silenced in apple, we get early flowering in apple as well. So again, there is some indication that this gene is responsible for inhibiting the flowering transition, and we by lowering their activity, the activity of TFL1, we induce flowering. TFL1, on the other hand, positively regulates vegetative growth. So it is a linear response. We get more vegetative growth by high expression of TFL1, and again at the cost of flower transition. Now the question is TFL1 one of the key genes for biennial bearing? And there is a lot of evidence, and Anius indicated I only have 8 to 10 minutes, so I will go through this very quickly. But TFL1, from the literature report, but again from some of our own work, seems to be regulated by a range of plant hormones, cytokinins, auxins, as well as gibberellins, seem to regulate the TFL1 gene. And indeed, when we look at the crop load in apple, TFL1 seems to be highly regulated in high cropping trees, and that might already be one indication that TFL1 is in fact the gene or one of the key genes that inhibits flowering under high cropping situations. Let me very briefly discuss the relationship between GA and TFL1. We did some in situ hybridization work. This is where we try to locate the gene inside the meristeme. And we did this for the repressor gene TFL1 as well as for GA20 oxidase. And GA20 oxidase is a gene that is involved in the late steps of GA synthesis, biosynthesis. And we compare on and off trees. And as you can clearly see, both genes are more highly expressed in the on trees compared to the off trees. So there might be some inhibitory effect of GAs involved in flower induction. When we look at the TFL activity in relation to the crop load over time, you can see that we have much higher TFL1 activity in high cropping situations compared to low crop. Again, some compelling evidence that TFL1 is a gene that inhibits flower induction. Crop load, since crop load is important, here are some figures from our own work where we look at the yield in the subsequent year of flower thinning, flower and fruitlet thinning. And there are two key messages coming out of this, this figure. The take-home message from this figure is the earlier the thinning is done and the more intensively the thinning is done, the better the subsequent yield in the next year, in the following year. You can clearly see here that the dotted line where we only left flower clusters 30 centimeters spread apart, we have a thinning intensity of 75%. Here we get over time the best yields in the subsequent year. Now, how do we want to go forward? We initiated a big experiment this year where we're looking at Spencer seedless. Spencer seedless is not a commercial apple variety, it's an old English variety, but it's a fantastic cultivar to study biennial bearing in apple. It is a parthenocarpic fruit, meaning it doesn't set seeds, because you see here the flowers don't have petals. So it's petalless, and the bees are not pollinating, are not attracted by the flowers and not pollinating the flower. This cultivar is non-biennial. It's a strongly non-biennial cultivar. However, when we hand pollinate the cultivar, it sets seed and it becomes strongly biennial. So to have that difference between seeded and non-seeded fruit, biennial and non-biennial behavior within the same cultivar, it seems to be a very attractive model cultivar to study biennial bearing. Now, it gives us the opportunity to look at the signals that come out of the fruit with and without seed. And remember what I said earlier on, those signals coming out of the fruit may inhibit or promote the transition inside the bud. So we're looking at the molecular and the biochemical changes occurring inside the bud in relation to those signals coming out of the fruit. Well, just to give you a quick wrap up and a summary, the integrator genes FT and full, they seem to positively regulate flower induction, not only in apple, but also in many other plant species. On the contrary, TFL1 seems to be a strongly repressor gene for flower induction, but it positively regulates vegetative growth. So there's more growth response of leaf and shoot than the flower and fruit response. We continue this work, and we wanting to look at the further inhibitory or examine the inhibitory effect of TFL1 and the other repressor genes SOC1 and plant hormones in relation to biennial bearing. Particularly the work on Spencer seedless. Now, in the end, we hope that we can identify some candidate genes which can be used as molecular markers for future breeding activities, so we can develop new apple cultivars which are not biennial. But we also hope that we can identify and develop some specific compounds, like, for example, the FT protein we applied in the field, but unfortunately that was unsuccessful. But we might come up with other compounds we can apply under field situations where we can modulate the flowering behavior, and we can modulate and affect the amount of buds that turn into floral buds. And with this, I thank you very much for your attention, and it's been my pleasure to talk to you from Germany, and I hope the video conference was fine. You could understand me well, and I'm very happy to take some questions if there are any. Thank you very much.
Thank you, Jens. Yes, that was a good feed, so that was fine. So, are there any questions for Jens about the project or the concept he's been talking about to start with? Okay, Jens. So the genes you were mentioning, are they corresponding to the actual biennial bearing varieties such as Jazz, Fuji, and the non-traditional biennial bearing Pink Lady? Is that what you're finding as well, or is that from the studies?
Well, the biennial bearing behavior is seems to be very common in the plant kingdom, and we see some a similar situation in annual plants as well as in perennial plants. So we believe that the mechanisms, the underlying mechanisms, whether it is at the molecular level or the biochemical level, seems to be more universal than we think, or than we originally thought. And we don't think it's a cultivar specific phenomenon. So most of our results and most of the results from the literature seem to be applicable irrespective of the cultivar. So whether you use a Jazz or a Braeburn or Gala, either they are biennial or non-biennial, and we can apply the results irrespective of the cultivar.
Okay. Just one question. I'll just pass the microphone to someone. Jens?
Thank you, Jens. Hopefully you can hear me. It's Heidi Parks from DAF Queensland. I'm interested in whether you've got any information or there's any information out there in the literature about the impact of temperature on the expression of those genes involved in that transition from a vegetative to a floral bud. So hot temperatures or cool temperatures?
Yes. This is an interesting question. Thank you for that. Now it is very, very difficult to look at the temperature effect under field conditions. We really would need to go into controlled environment rooms to look at the temperature effect separately from the light effect, because temperature and light are often very, very much related. When you have high light intensity, you tend to have also higher temperature. What we do know is...
Just go back 2 seconds of what you're saying. You just had a bleep. Sorry.
Say again?
You just cut for a brief second, so missed what you just said.
Okay. Okay. So just to go back to what I said, the temperature effect is very difficult to separate from the light effect under field conditions, because when temperatures increase, you tend to have also higher light intensities within the orchard environment. So you will need to go to conduct some experiments under controlled environment in the climate rooms. However, what we know, for example, the FT gene has a very strong diurnal pattern. It seems to be more highly expressed during the daytime than during the nighttime, and it has the highest expression at somewhere around the midday. And this might be correlated with temperature, but we don't know yet. We don't know yet for sure. But I think that the temperature has a very strong effect on the expression of some genes.
Well, I think that's all we've got on this end, Jens, for questions. So that was very informative on
yeah, and if you allow me, just one further comment.
Yep.
Just to be sure, we are not interested in developing transgenic apples. What I showed you, well, this is important for the audience. This is not for our for customers, our fruit growers, and in fact the legal situation in Germany is that we would not be able to introduce transgenic plants, transgenic apple trees into the commercial environment. The work we have done is really just to have a proof of function of the genes we are interested in, to understand what those genes do inside the plant. And when we understand the plant, the gene, and what it triggers inside the plant, then we can start manipulating those genes by other means, or we can we are sure that those genes are potential candidates for molecular markers for breeding activities.
One more question.
Thanks, Jens. My question is, it's probably a little bit more simplistic. It's along the lines of, we know we've got an on year or an off year, plenty of vegetative growth or not many fruit, but those buds at that potential flowering dates, 39 to 53 days after full bloom, we know it's initiating. Are there compounds out there that will suppress these genes so as it becomes less, you have less floral buds sitting there waiting for the next season because you've got so many of them potentially that will initiate into floral buds. So you've sort of got a crop regulation happening at that period. Do you understand?
Well, yes. Crop regulation at that period, or preferentially earlier on. If you wait 39 to 53 days after full bloom, you might be too late. If you know this is a strongly biennial cultivar, and that cultivar is in an on year, that cultivar should be thinned down at best at full bloom or soon after full bloom. And as one of the slides I showed, the earlier you do it, the better it is for the subsequent year. And if you do this, and this might be some of the compelling evidence that the gibberellins and auxins inside the fruit are involved, that if you do it early, fewer inhibitory signals coming out of the fruit and the suppressing effect on subsequent on the following year is less.